Well, first of all, yeah, thanks for having me. It's a real privilege to share our work with you guys. And yeah, I hope you guys are interested in these molecular quantitative trait loci and context. We've been having a fun time exploring these in the lab with my colleagues. So we're in UNC Chapel Hill. Like Sarah said, you can check out a little bit more about our lab at the URL shown here. And yeah, I'm Bran. People call me Bran, and I'm a fifth year PhD candidate in the bioinformatics and computational biology program. And so I've made the title a little bit shorter and sweeter here, but a more descriptive title is on this preprint that Sarah mentioned that you can check out. And so I'll be talking to you guys today about how we've used Wnt activity to reveal context-specific genetic effects on gene regulation in human neural progenitor cells. All right. So one thing that we've learned from genome-wide association studies is that common genetic variants, those variants that are present at about 1% or greater in the human population, these variants impact brain traits. So here I'm showing a GWAS for structural brain traits that are measured via MRI in adult cortical tissue. And what we can see here is that this Miami plot is on the top is showing the results from the GWAS for cortical surface area. And the bottom of this plot is showing the GWAS results for the thickness of the cortex. And just to remind everyone what these plots are showing, we're laying out the genome along the x-axis with the chromosomes labeled here. And each dot represents one of these common genetic variants that may differ between people in the human population. And the height of these dots on the y-axis denotes their significance. And so you can see for both surface area and for thickness, there are genome-wide significant variants that seem to impact the surface area and thickness of your brain. And not just structural traits are impacted by common variation. There's also complex functional traits like the risk for autism spectrum disorder. So here's a Manhattan plot from a recent GWAS for ASD. And similarly, you can see that there are genome-wide significant variants that kind of come up above this genome-wide significant line. And that we can identify what these variants, where they are, but we really don't know from GWAS what they're doing. It happens that all these variants fall mostly in non-coding regions of the genome, where it's really difficult to assign a specific function to what that variant's doing that could impact these traits. So this is a major question that I think that GWAS results can't really get at. So one thing that we can try to do to understand how different genotypes that you can inherit, here I'm just using an example of an allele that can come as a C or an A. And we know from GWAS studies, like we just saw, that maybe the A allele could be associated with increased risk for a neuropsychiatric disorder or some kind of brain trait. So what's going on in the middle here to actually convey that genetic risk? Well, one thing that people have tried to do is to look at these intermediate things that could be leading to disorder risk. And one thing that we would expect is if these variants are in non-coding regions of the genome, they're not affecting the composition of proteins necessarily, but so what might they be doing? Well, non-coding parts of the genome are ostensibly regulatory in their function. So we can imagine that perhaps this A allele may be associated with chromatin accessibility, such that if you inherit two copies of the A allele, you have an accessible region of the genome. And maybe if you have the two copies of the C allele, this regulatory element is relatively closed. And we can kind of use this chromatin accessibility as a proxy for transcription factor binding. If the chromatin is open, we could expect that there may be transcription factors that could accumulate and bind that DNA and exert some sort of regulatory function on gene expression. So when we find genetic effects on chromatin like this, we call them chromatin accessibility QTLs. We can also look at genetic effects on gene expression. We call these expression QTLs. And these, so in this case, in this cartoon example, the A allele here may be associated with increased transcription from this nearby promoter. And so we can try to overlay the genetic effects on these molecular phenotypes with genetic effects on a complex brain trait like risk for neuropsychiatric disorder. And we could try to kind of build a hypothesis where the same variants that are conveying disorder risk may be doing so by acting on specific regulatory elements and genes. So we've mapped a lot of EQTLs so far, and traditionally these EQTLs have relied on adult post-mortem bulk tissue samples. And there's been a lot of EQTL discovery using those kind of approaches. But if you try to explain GWAS loci this way, there's some disappointing inability for these EQTLs to really explain a mechanism. So what this figure is showing is the percent of independent GWAS loci that are explained by EQTLs in adult brain tissues. So, we're looking at the vast majority of GWAS loci remain unexplained, at least by these gene expression EQTLs in this sort of bulk postmortem adult tissue. So where is all the rest of this heritability for these disorders hiding? People in the field have been calling this recently missing regulation. These variants seem to be exerting an effect that we haven't fully understood yet. So where might this stuff be hiding? So two possibilities are that genetic variants will only exert their effects on gene expression or chromatin accessibility in certain cell types and in certain contexts or stimulated states. So let's take first the example of cell type specificity. So imagine here a variant that could be either A or C. And if this variant is in a particular cell type on the bottom, perhaps this cell type doesn't express a transcription factor that would bind to this site. And so there's no real effect on gene expression because this cell is not even really expressing this gene, not expressing the regulatory factors that are necessary to interact at this site where the variant is. But maybe in a different cell type, there are transcription factors whose binding could be altered by this C allele. And we could then uncover this missing genetic effect by looking in particular cell types that are relevant to a trait of interest, perhaps. So for our study, we tried to address the cell type specificity by looking at molecular QTLs in human neural progenitor cells. And so just a little bit about these cells. So these cells come from the fetal dorsal telencephalon, which is tissue that goes on to give rise to the cerebral cortex. And these are dissociated from fetal tissue that comes from roughly gestation week 14 through 20. And we have about 82 of these genetically distinct lines that are all genotyped. And these cells are radioglia. They express these proliferative radioglia markers like SOX2 and PAX6. And these cells are really powerful because they kind of sit at the ventricular zone where they are exposed to a lot of developmental signals. And these developmental signals can guide them to either produce symmetric cell divisions that produce more radioglia that could potentially expand the resulting cortex that they generate. And then they also can produce asymmetric divisions, which can produce these immature neurons or intermediate progenitors that will then go on to become neurons later, that then climb towards the surface of the cortex to make this layered cortical architecture. So these cells are the ones that we thought would be interesting to look at molecular QTLs within. And just on the right, showing you that the genotypes that these 82 cell lines comprise, they span a variety of different genetic backgrounds that come from a whole bunch of different ancestries. This genotype diversity is something that is, I think, really cool about this resource, especially when a lot of genetic effects are typically isolated to European populations. So are these are the regulatory elements that are active in these cells, are they actually important for neuropsychiatric disorders and brain development? Well, we can try to evaluate that and see if these are a good cell type to look in by looking at partition heritability. And in partition heritability, what we're trying to do is understand how much of a genetic signal could be enriched within some annotated portions of the genome. So in this cartoon example of how partition heritability may work, what we have here is a zoom in on a particular locus that's associated with some brain trait. And just like a Manhattan plot, each dot is a variant and the significance of the variant is plotted on the y-axis. So say there's a significant locus here associated with, let's say, autism risk. And we can look to see whether that signal falls closer to certain regulatory annotations that we can define. So here we're looking at two different kinds of elements, one that are active from brain tissue and maybe another from non-brain tissue like the liver. And so here we could say that the heritability for this brain trait seems to be concentrated more towards these brain regulatory elements, more so than these liver elements. So by using partition heritability, we can kind of see the genetic contributions that are shared between this GWAS trait and where those genetic contributions fall in annotated regions. So neuroprogenitor cells show a strong enrichment of heritability for lots of neuropsychiatric disorders. And so this is a plot of the enrichment that we get in either the progenitors versus neurons. So a former student in our lab, Dan, differentiated all these cell lines into neurons and compared partition heritability with those differentiated cells and compared back to their progenitor counterparts. And we found increased enrichment of heritability of all of these different neuropsychiatric disorders in progenitors. So a lot of risk for these traits may start quite early while these cells have yet to become neurons. So another thing that we can do to try to find some of these missing genetic effects is to consider specific contexts that the cells may find themselves in and specific stimuli they may be exposed to. And the reason why these could reveal new genetic effects is, again, kind of thinking about how transcription factors could be context sensitive. You could imagine a variant here in a regulatory element, either A or C alleles. And up here, maybe the cell is in this resting state where there's no signaling activation and transcription factors are sequestered away from this regulatory locus. Maybe upon stimulation, however, this transcription factor is able to translocate into the nucleus and interact with the A allele, but not the C allele. So in this stimulated state, we may uncover a new genetic effect that was invisible without the stimulation. So what kind of stimuli do we want to use to find these context-specific molecular QTLs? Well, for this study, we focused on the Wnt pathway. And on the left, I have a cartoon of the canonical Wnt signaling pathway. And just to walk you through it briefly, it starts at the cell surface where there's a membrane-bound receptor called frizzled, and Wnt ligands will bind frizzled. And what that will do is it will destabilize this destruction complex that's made of axin and some other partners. And when this destruction complex is destabilized, it can no longer phosphorylate and target beta-catenin for destruction. And as beta-catenin accumulates, it's able to translocate into the nucleus and interact with other transcription factors like TCF-LEF and drive the transcription of Wnt target genes. And we thought the Wnt pathway would be a really good one to study for a number of reasons. And one is that if you look at GWAS loci for differences in cortical surface area, similar to that first Manhattan plot I showed you, if you look at the genes that surround those loci and you run them through a gene ontology analysis, you can see that some of the most enriched categories are Wnt receptor signaling type things. So one piece of evidence that Wnt seems important for the surface area of an adult brain. And furthermore, there's a lot of other interesting results through the years that show Wnt is really important in brain development. If you make a form of beta-catenin that's constitutively active and in a mouse, this can cause this sort of really shocking amount of cortical expansion to the point where the mouse develops these gyri, kind of what you would maybe think of in a primate. We know that Wnt has roles in maintaining neural progenitor cell potency, regulating their timing and extent of their differentiation. And we also know that if you take cell lines that are derived from people that have neuropsychiatric disorders like schizophrenia, these types of cells show different Wnt signaling output. Same for autism. There's many Wnt components that have been identified as potentially important in autism. So with our cell type of neural progenitors in hand and our pathway of Wnt stimulation selected, we were hoping that we could map QTLs and find some of this missing regulation that could better explain GWAS traits. So here's how the experiment happened. And so what we did is we took our genotyped neural progenitor cell lines, take about 8 to 10 of these lines per week, and we'll expand them for 3 weeks in order to get enough cells for these downstream sequencing applications. And during the last 2 days of this expansion, we exposed them to either vehicle treatment or Wnt stimulation in 2 different ways, which I'll explain a little more about in a second. And I just wanted to highlight here that this cell culture work was, Jordan here is instrumental to getting all of this done. It was a big undertaking to grow up all these cells and treat them and then capture their molecular profiles. And so he was super helpful and worked closely with him during the pandemic to do all of this. So with those cells exposed to these either vehicle or Wnt stimulating compounds, we then captured, we took cells for ATAC-seq and total RNA-seq. So kind of like the cartoons I've shown before, we were hoping to discover variants that affected the accessibility of chromatin regions that may only exert effects in Wnt stimulated states, same for gene expression. And on the right, what we're hoping to do with these QTLs is better explain GWAS traits. So I'm showing 3 kind of cartoon versions of a locus zoom plot where we're looking, zooming in on a GWAS signal with the significance again plotted on the y-axis, and genomic coordinates would be along the x. So say we have a GWAS trait of interest where there's a significant association. We can overlay our molecular QTL data. Here, maybe this could be a chromatin or a gene expression QTL. And if you just look at the vehicle or baseline QTL, maybe we are not able to co-localize this GWAS signal with our QTL signal. But upon Wnt stimulation, we see a significant effect on, let's say, gene expression arise. And so this can help us identify if we can look at annotations around this region and see if this can help us pinpoint Wnt-responsive regulatory elements, promoters, and target genes that could potentially be part of a causal mechanism leading from genetic variation towards the expression of some GWAS trait. And before I move on, all of this kind of the analysis and bioinformatics that went into this work, really pivotal contributions from Nana Matoba, who I think may be tuning in all the way from Japan right now. Really, a really great guide and mentor for me learning a lot of the statistical genetics and bioinformatics that went into this work. So thanks to both of those people. All right, so here's how we stimulated the Wnt pathway. So on the left of this cartoon, Wnt is off. And on the right side, Wnt is on. And so we stimulated the pathway 2 different ways. One with Wnt3A, a recombinant ligand that more or less mimics what you could expect from endogenous Wnt signaling that starts at the frizzled receptor from outside the cell. And then the second way we activated the pathway is through this small molecule called CHEER. And CHEER is a really one of the strongest known activators of Wnt signaling, and it works by inhibiting the GSK3 beta enzyme, which is the phosphorylase that's part of this destruction complex that normally would target beta-catenin for destruction. So by inhibiting it, we accumulate beta-catenin, and once it translocates into the nucleus, in the Wnt on state, we would expect it to associate with TCF left type transcription factors at these Wnt regulatory elements. So we could expect that when Wnt's on, we may find open regions of chromatin that harbor lots of these TCF left transcription factor binding sites that beta-catenin could be interacting with. And again, in the off state, these would be more inaccessible when beta-catenin is not there. So let's take a look at what we found out about how the the chromatin accessibility across the genome changes when you stimulate with Wnt. And it was really confidence building to see these results, to see that we actually got some expected transcription factor binding site enrichment within these Wnt-sensitive regulatory regions. So what these plots are showing, so we're looking at the enrichment of transcription factor binding site motifs within these changing regions of chromatin. So to the right are the regions that open in response to Wnt, and a smaller number of these closed, and these are depicted to the left of these plots. And the significance of the enrichment of the transcription factors is plotted on the y-axis. So here, Wnt3a stimulated changes in chromatin accessibility. Here, CHEER stimulated changes. And in both of these conditions, we found some of the major players of that would mediate Wnt activation, like TCF7 and LEF1. These binding sites were highly enriched, or among the most enriched in these regions that we found. So this gave us some confidence that we were actually doing something interesting to the genome in an expected way that is consistent with Wnt signaling and what we know about it already. So switching over to gene expression, similar plots showing the changes in genes as a result of stimulating the Wnt pathway. Again, Wnt3a on the left and CHEER on the right. And so if you see the log fold change above 0, this means that the Wnt stimulation increased the expression of that gene, and below 0 meaning Wnt reduced the expression of that gene. So each one of these dots is a variety of is a gene, and we've labeled a variety of them that are relevant to Wnt stimulation. And so, kind of similar to the chromatin regions that we found some sensible genes that changed in reaction to Wnt stimulation. So lots of different genes changing, and happy to see that there were some known targets of Wnt stimulation that were among them. So we can also take a combined look at the chromatin accessibility peaks and the genes nearby to see if we can find gene regulatory relationships where perhaps a regulatory region could accumulate transcription factors, interact with a promoter of a nearby gene to drive its expression. So what we're looking at here is a locus for axon 2, one of the components of the Wnt signaling pathway, a major player in that destruction complex. And so we're centering the plot here at the promoter of axon 2, where you can see there's a strong chromatin accessibility peak that is visible both in vehicle condition and also in Wnt3a stimulated conditions. So these plots are showing you the attack-seq sequencing read depth across this locus. So there's, by eye, there's maybe only subtle changes in chromatin accessibility that you can pick out with the plots depicted like this. But what's really interesting is we can take the accessibility in this region and correlate it to the expression of axon 2. And we found that there's only significant correlation of nearby peaks with axon 2 expression in the context of Wnt3a stimulation. So under vehicle, the promoter not correlated with accessibility nearby, but under Wnt3a stimulation, this promoter becomes strongly correlated with multiple chromatin regions in this area. So showing that we can uncover new gene regulatory relationships under stimulated states that you would miss otherwise at baseline. And we can try to look at some other things that are going on in this region to kind of make sense of it and maybe develop some sort of mechanistic hypothesis. And one way we can do that is by looking at transcription factor binding sites again within these near the promoter and within these chromatin regions. And so here you can see there's a lot of these TCF7 and LEF1 motifs that can be found both at the axon 2 promoter and in some of these highly correlated peaks nearby. And we can do that with predicted transcription factor binding sites, and we can also do that with functionally defined transcription factor sites that we learned through ChIP-seq experiments. Great. So with all of these changing chromatin regions and gene expressions in hand, we wanted to see whether these were actually relevant to complex brain traits. So taking the differentially expressed genes first, we tested for enrichment within the set of genes with curated disease-associated genes from this DisGeneNet database. And just showing up for a subset of the gene categories that we tested here that are relevant to neuropsychiatric disorders. So we're looking at schizophrenia, autism, bipolar, and major depressive disorder here. And for each of these categories of differentially expressed genes, or maybe not differentially expressed genes at the top, here we have the Wnt3A changing genes, all of them, or just the ones that go up or down, similarly down here for CHEER. We can see that, that there are enrichments of these disorder-associated genes for both Wnt3A and CHEER, and there's some suggestion of specific effects from the CHEER up, the genes that went up in response to CHEER treatment. And in contrast, if we look at the genes that do not change in response to Wnt, these do not show enrichment with these curated disease gene sets. So it helps us understand that that Wnt stimulation is affecting genes that are relevant for neuropsychiatric disorders. Turning back to the regulatory regions that change in response to Wnt stimulation, we can look at the enrichment of heritability for a variety of GWAS traits within those regions. So here, this is going back to that method I showed a cartoon of earlier, partitioned heritability. So what we're showing here are various GWAS traits along the x-axis, and then various categories of regulatory elements along the y. So it's been shown already from other studies that mapped regulatory elements in fetal brain that these kind of developmentally active regulatory regions are strongly enriched for risk for basically all of these neuropsychiatric disorders, including some cognitive traits too, but not so much for neurodegenerative traits, and not so much for non-brain traits. So this yellow bar shows a category of GWAS traits that are not brain related, like IBD, asthma, rheumatoid arthritis, and things like that. And if we look at specifically regulatory elements in progenitors, not just the bulk fetal brain, you can also see some of these enrichments across these categories. And from our study, we took the regulatory elements that changed in response to Wnt or CHEER, and we saw some strong enrichment of heritability, especially for these neuropsychiatric disorders and these cognitive traits. Didn't see enrichment for neurodegenerative things, which probably disease mechanisms for those may occur later in life. So we did not see enrichment there, and we didn't see enrichment for traits that are not brain related. Just to highlight before I move on from this slide, we also see that these regulatory elements show enrichment with variants that affect the thickness and surface area of the brain. And kind of focusing on just on those structural brain traits for a second, we can look at the regional specificity of how this heritability enrichment is being harbored within these regulatory elements. So on the top, I'm showing a diagram of the brain and highlighting the region regions where we see an enrichment of variants that have to do with surface area within these Wnt-sensitive elements, and down here for thickness. So some of these regions show enrichment under both Wnt3A and CHIR, and some of them showed specific enrichments to one or the other of the stimuli. So together with all this enrichment analysis of heritability, we can see that these changing regulatory elements, they seem to be relevant to brain traits. So from here, we wanted to map genetic effects and on both chromatin accessibility and gene expression. So we wanted to get at QTLs. And since we have this context specificity to our experimental design, we were also able to identify a special category of QTL that we're calling response QTLs. So again, take this example genotype that comes in either a C or an A allele. And we're going to capture all these attack-seq profiles and look to see changes in chromatin accessibility. And if this genetic variant is associated with changes in that accessibility, we call that a chromatin QTL. We may find that only under Wnt-stimulated conditions do some variants actually exert their effects. Maybe they become detectable only after that stimulus. And whether that's on chromatin or gene expression, when we see this change in genetic effect, this interaction between the condition and the genetic effect, we can call this a response QTL. So to break that down a little bit more specifically, here's what we can, some examples that help understand what these QTLs and response QTLs may look like. So here I'm plotting the counts of either attack-seq or RNA-seq on the y-axis, and then along the x, I'm looking at the number of alleles of a particular QTL for a single variant, either 0, 1, or 2 of the minor allele. So there's going to be a lot of variants that do not show a genetic effect, which are not QTLs, where we see this flat line. It doesn't matter how many alleles you have of this variant, you still have basically the same amount of sequencing reads. And we may see that the read count goes up in the context of Wnt stimulation because this is potentially, let's say, a differentially expressed gene, but this is not a QTL. Wnt effect, no genetic effect. But we can also find QTLs where we see this additive effect of this allele. And in this case, I'm showing you an example where there's genetic effects, but there's really no difference in the effect with or without Wnt stimulation. We can also see cases like this where there's both a genetic effect and an additive effect where the line is shifted up by the Wnt stimulation context. So this would just be, this would still be a QTL, but does not show an interaction between genetic and condition effects. So lastly, here's a response QTL where under baseline conditions, we see no genetic effect. And under Wnt stimulated conditions, we now see a stronger genetic effect. So basically, when we see a change in slope that's significant between the baseline and the Wnt stimulated context, we can infer a response QTL. So just to quickly show you broadly the results from this QTL mapping, we found a lot of genetic effects on chromatin accessibility, which I'm showing you here. So we found tens of thousands of variants with significant effects on chromatin in both Wnt3A and CHIR stimulated conditions. I'm showing these as Miami plots, where the Wnt-specific stimulated conditions are depicted on the top half, and the baseline or vehicle context is shown on the bottom half of these Miami plots. And we've highlighted a few interesting genes that were of note from some downstream analyses when we compared these to GWAS that we can talk about a little bit more about later. But also I'm pointing out that these circled dots, I know they're very small, these circled dots are the response chromatin QTLs that we found. So a subset of the QTLs we find show these response dynamics. Lots of genetic effects on chromatin, and lots of genetic effects on gene expression too. So switching over here to the eQTL side, not quite as many for sure as the chromatin ones, but we found a couple of thousand significant EQTLs for Wnt3A and shear conditions. So I know these Miami plots are not super informative, but let's dive into like what we can gain from these context-specific approaches. Before I do, just want to make sure that we are confident that these genetic effects are real and robust. So one thing we did to do that is we checked to see if we were reproducing effect sizes for each SNP compared to previous studies in the lab. So what I'm showing here is scatter plots where we're plotting the effect size of a variant from our study on the x-axis, and this is taken in vehicle conditions here, compared to other baseline QTLs that were previously mapped here from Dan's paper, looking at chromatin QTLs in our progenitors at resting baseline state, and here looking at eQTLs from Nil's paper. And we see a really strong and significant correlation of effect sizes. So we think that these genetic effects are believable, and in the sense that they show really strong reproducibility with previous data. So one thing we can look at here is how much more genetic effect can we identify in these stimulated states. So one way we can look at this is using these upsetter plots. And if you haven't looked at these before, they're sort of like deconstructed Venn diagrams where we have different categories listed down below, either vehicle or either of the Wnt stimulated states. And on the top, we're looking at the number of, in this case, genetically regulated chromatin peaks that are discovered in each of those categories. So for example, we found a lot of genetic effects on chromatin that are shared regardless of the condition that you map them in. But in red, we were able to find these Wnt-specific genetic effects on chromatin. In fact, we found a 66% increase in the number of genetically regulated caPeaks when we in the Wnt stimulated state compared to vehicle. So on the right, same idea, but for gene expression, we're able to find about a 50% increase in the number of genetically regulated eGenes in Wnt stimulated states compared to vehicle. So we were happy to see this because I think it bears out sort of a suspicion in the field that's become more and more, I think, well supported, that looking at QTLs in these dynamic states are going to really afford you, are going to identify a lot of novel genetic effects. Another cool thing we can do with the combined ATAC-seq and RNA-seq data, we can look at the QTLs we map from it, and we can combine them to infer enhancer priming. And so enhancer priming is something we can see where you might have a genetic effect of this G allele, for example, that's present in a naive or unstimulated state. So we see the G allele opens the chromatin here at this site, maybe by allowing binding of a pioneering transcription factor. But at baseline or unstimulated conditions, we don't see an effect of this allele on gene expression at all, even though it may be open for business on the chromatin. Now, when we apply some kind of stimulus, now we see that we uncover this allelic effect on gene expression that we didn't see before, potentially because the stimulation generates a key transcription factor that now is available to participate in binding at this previously open site and now drive the expression of a target gene from a nearby promoter. So we looked for this combination of effects on chromatin and gene expression to infer enhancer priming within our data. And we found a number of primed loci. And I'm just going to show you one example of this here at the Clint1 locus. So kind of what I was just describing, you can see these locus zoom plots where, again, each dot is a variant associated with either chromatin or gene expression, and the significance on the y-axis here. And at this region on chromosome 5, we see chromatin accessibility that is effects on it from these variants that are present in both vehicle and cheer conditions, but only an effect on gene expression under cheer stimulated context. So we can use this to kind of infer a potential mechanism how these variants can work that includes both of these molecular phenotypes. And down here, we'll look at some more of these plots later. So just to kind of make sure you understand how they're shown, down below these, the association data, zooming in on this chromatin accessibility peak to show the pileup of ATAC-seq reads and show kind of the difference in accessibility you can see based on the different genotypes for this lead variant shown with this purple diamond. And we can look at annotations as well within this peak and notice that there are Wnt-sensitive transcription factor binding sites within it. Again, these left TCF7 binding sites are present in this peak. So we can plot genetic effects on chromatin and gene expression for this lead variant at the CLINT1 locus. And here we see that in both baseline states in gray and the cheer stimulated in blue, we see genetic effects on chromatin accessibility. And we see this kind of change of slope in the effect on gene expression. So we can develop this potential, this mechanism where the G allele at this site is maybe more conducive to binding of a transcription factor that maintains the openness of the chromatin here, but only upon Wnt stimulation do transcription factors that will drive gene expression arrive. And in this case, they may be TCF-LF elements or factors based on the presence of that binding site in the peak. And we thought this was an interesting example because CLINT1 is a protein that could potentially affect Wnt signaling itself through its roles in endocytosis and secretion of Wnt ligands. So another thing that we looked at that we thought was really cool is we can look at all of these genetically regulated chromatin regions, and we can overlay them with various genomic annotations. And one thing we looked at were regions of the genome that have been identified as being part of this human-specific evolution along the lineage between diversification from chimpanzees and humans. And so there's two types of annotations we considered here: HARs, human accelerated regions, and hackers, human ancestor quickly evolved regions. And so these are distinct but sort of similar annotations. HARs show regulatory elements that could have had some ancestral function, but then changed and gained function potentially after the divergence with our chimpanzee cousins. And hackers, similarly, these are regulatory elements that were ostensibly non-functional before we diverged from chimps, but along this human-specific lineage may have gained some new human function. And so these regions are all defined by just species sequence comparisons. And some of them have been shown to have some interesting functional results. So this one HAR, I think it's HAR E5, is a distal regulator of the gene FZD8, frizzled 8, one of the Wnt receptors. And if you take the human-specific form of this enhancer or the chimpanzee-specific form of the enhancer and you transgenically express it in the developing mouse brain, only the human-specific form shows this effect where it reduces the cell cycle length of neural progenitors and results in the mouse having a larger neocortex. So really kind of interesting and suggestive that some of these evolutionary relevant regions may actually be driving some of these human-specific features, like having a really foldy and expanded cerebral cortex. So we overlapped these HARs and hackers with our regulatory elements, and we found that we could detect a whole lot more overlap when we considered the Wnt-stimulated states. So here on the left, we're just showing the significance of the enrichment of the various categories of genetically regulated chromatin peaks. But I think maybe the easier way to look at this are these upsetter plots again. We can find these new overlaps with HARs on top and hackers on the bottom in these Wnt-specific states. So implying, I think, that these regulatory regions that I think mostly have not been explored functionally, these regions that may drive human evolution in some way, they harbor genetic variation that is Wnt-sensitive and can affect the local chromatin accessibility. So we thought that was really neat. And just to show you one example that I mentioned a little bit already, we actually found overlap of variants that regulate chromatin accessibility within this HAR that is a distal enhancer of that frizzled 8 gene, the one that was shown to change the development of the mouse brain when expressed transgenically. So we thought this was really cool, and we can zoom in on the peak that is regulated by this variant right here, and note that it also has some of these Wnt-sensitive transcription factor binding sites within it. So yeah, really excited to see that some of these Wnt-sensitive elements may be relevant for human evolution. Last thing that I'll talk about is how these genetically regulated peaks and genes, they offer new opportunities for explaining GWAS loci. So here we're looking at LD-based overlap of our QTLs with GWAS. And so on the left, we're looking at the total number of peaks or eGenes that we consider that we found in our study. And in the solid colored bar down here, we're looking at the number of those peaks that actually showed a close LD supported overlap with a GWAS. And we considered lots of GWAS traits, mostly structural brain traits and neuropsychiatric disorder GWAS traits. And you can see on the right, if you look at the unique number of colocalizations between these QTLs and GWAS traits, as we load in more context specificity, we can increase the number of colocalizations we find, especially for these genetically regulated chromatin peaks. And again, upsetter plots, I think, show this better. But you can see here for chromatin accessibility and over here for gene expression, that we're able to find a lot more overlap with GWAS when we consider these Wnt-stimulated states. And we looked for the gene expression side. We wanted to know how, like, kind of the novelty of these overlaps. And we noted that, like, just looking at GTEx bulk post-mortem QTLs from the frontal brain cortex, so a relevant tissue type from GTEx, that about 14% of the eGenes we found that had overlap with GWAS signals, that these were not previously reported as eQTLs and GTEx. So we're uncovering some new opportunities for explaining GWAS. So here's an example of that at the ANC-RD44 locus. This locus on chromosome 2 has been associated with both schizophrenia risk and the volume of a part of the hippocampus. So here we're showing the signals for those GWAS on top. And below we found overlap with this response QTL that shows the specific effects in the cheer stimulated state. So no effect on gene expression in vehicle, or at least not significant. And under cheer stimulation, we see a significant effect on gene expression on ANC-RD44. So at this locus, we could start to understand that potentially the effects of these variants on these complex traits may be mediated through ANC-RD44. And in the way that perhaps the T allele that these GWAS said is associated with lower schizophrenia risk and lower hippocampal volume, that the same T allele is associated with increased expression of ANC-RD44. And interesting to neural progenitor cell biology and Wnt signaling, because this protein is known to have roles in cell cycle regulation and also can suppress signaling of pathways that have crosstalk with Wnt, like the NF-kappa B pathway. Okay, I'm going to try to wrap up quickly, and so I have a little bit of time for questions if you have them. So just to kind of summarize the results from this preprint, we're able to address and find this missing regulation that bulk adult post-mortem QTLs have not been able to discover in the past by considering cell type specificity and doing QTL mapping within a homogeneous population of neural progenitor cells, and by considering the activation of a developmentally relevant and also disease relevant pathway like Wnt. So we think that this is a promising example of how we could do this for other pathways too. I'm just kind of putting up a bunch of text that kind of walks through the bullet point wise what we've talked about already. I'm not going to read through it, so I can give you a little bit of a peek at some of the things we're doing in the future. So since this worked well for the Wnt pathway, we would love to explore similar experimental designs looking at different stimuli. So you could definitely do different developmental signals like Notch, Sonic Hedgehog, and things like that. But you can also try to do this with clinical stimuli. And we've done this so far, started doing this with treatments like lithium and valproic acid, which can be prescribed to people with bipolar disorder. And we can do this and try to look at genetic effects on gene expression and chromatin that may have pharmacogenomic relevance. We can look at whether these loci are also associated with the clinical response and try to understand what regulatory elements and genes may be affecting people's difference in how they respond to these clinical treatments. And additionally, not just molecular QTLs can help understand all of these complex traits. We also would love to consider cellular pathways that may be downstream of the gene expression and chromatin themselves. So we've done this by trying to do cellular QTLs, where we look at genetic effects on the proliferation of these neuroprogenitors, as well as their capacity to respond to Wnt, as measured by like a luciferase reporter assay. And if you want to see some results on that, we put out a paper that came out early this year that shows some genetic effects on lithium-sensitive proliferation of these cells. So yeah, we hope that trying this with these clinically relevant compounds could help understand some of the variance in clinical outcomes, like for lithium or valproic acid specifically. And we're hopeful that this can really aid this pursuit, because the previous studies that have tried to look at genetic effects on, let's say, clinical response to lithium, they found very few loci. They're interesting, and they show they can find genetic effects, but they're underpowered. It's hard to get enough people, a big enough sample size of responders and non-responders to these clinically relevant drugs to do these studies really well. And also the phenotyping of telling whether someone is a good or bad or not so good responder to a clinical treatment is sometimes subjective, and something that makes it difficult to do genetic association to. So we're hoping that by considering molecular phenotypes, cellular phenotypes, that we can kind of drill down at what are the underlying mechanisms that may be affecting clinical responses and responses to environmental insults as well. So I hope I've been able to convince you today that putting QTLs in context is a really good idea, that it's interesting to see genetic effects that change in response to your favorite signaling pathways, treatments, or environmental exposures. And we hope that we see a lot more ability to understand how GWAS loci actually work by putting them in the context of molecular QTLs. Thank you so much for your attention. And I just wanted to end by thanking my lab. It's been a really, really great place to learn how to be a scientist. Jason Stein's been an excellent mentor with really cool ideas that help guide this project. And just highlighting in particular one more time, Nana and Jordan, who I worked closely with and are my co-first authors on this paper, on that preprint, that I couldn't have done any of this without them. Also, a bunch of really great collaborators, thesis committee people and core facilities at our university that have been really instrumental in getting all this done. And I hope I've left a little bit of time for your questions.